Hello everybody, today we're going to learn about the DNA damage and the different repair mechanisms that are used to repair the damage. Furthermore, we will talk about the ways in which researchers are utilizing this knowledge on targeting DNA repair mechanism to help uh, treat disease, including cancer. So what is a genome? A genome is an organism, genetic instruction, and is made of DNA. You're probably familiar with the seeing set of a chromosome, which is how an organism genome is condensed and packed. The human genome contains 23 pairs of a chromosome and a total of 46. The human genome is comprised of 23 chromosome pairs. You have a pairs of a chromosome because you inherit one from your mother and one from your father. In a total 23 chromosomes from each parent. Does every organism have 46 chromosomes? Absolutely no. So have many more, and some have just one, like bacteria, viruses. Every cell in your body contains a copy of your genome. That means every cell in your body has a full set of a chromosome. That's a lot of DNA. All the DNA in your body, some of the about half of a pound, and it is believed that is E. coli your DNA could stretch over 10 billion miles. That's all the way to Pluto and back. DNA is a long molecule composed of the combination of four nucleotides, ATCG. Your DNA encodes instruction for the work a cell should be done. It is like a language for your cell, except the alphabet it uses only has 4 letters. The nucleotides are the four small molecules: adenine, thymine, cytosine, and guanine, abbreviated by ATC and G. The DNA in your cell actually composed of 2 molecules of DNA held together by nucleotides pairing with each other. The two molecules are said to be complementary. The shape that they form when paired is a double helix. It is this structure that Watson and Crick defined it back in 1953. Do you know how the nucleotides pair together? DNA nucleotides pair up in a consistent way. Adenine always pairs with a thymine, and cytosine always pairs with a guanine. A pair of nucleotides in a DNA double helix is referred to as a base pair. So DNA sequence is the order of nucleotide of a DNA molecule. When we look at the double helix, we only need to know the sequence of one of the molecule. So why is this? That's because nucleotide pairing follows strict rules. We can always figure out the sequence of the complementary DNA molecule. So let's end with the DNA and take a closer look. If we end with one of the molecule and write out its nucleotides in order, we can now read the DNA sequence. Here is a DNA sequence. Based on what we know about the DNA-based pairing, we can go back to figure out the sequence of the complementary strain of the DNA. The genome can be divided into coding and non-coding regions. Of our entire genome, only 2% contain coding DNA. Genes are found in the coding region of DNA. Genes are the region of the DNA containing instruction for the cell to make a functional RNA or a protein. A protein is a large molecule that does a specific task in the cell. like breaking down cell waste or importing nutrients. It is believed that the human genome contains about 20,000 genes within the coding region of the DNA. So more recent information suggested that the number could increase to 40,000 genes when you take into consideration all coding genes and functional non-coding genes. One way to think about the role of the genome is like it is a cookbook for a cell. The whole genome is a cookbook full of the recipe for the cell. Having only 4 different types of the nucleotide doesn't seem like it would give you much variety. But think of how much the order matters in the letters in a book. If you keep all the same letters in a book but scrambled them, would you have the same book at all? The whole genome is the book full for the recipes for the cell. So that's one way you can figure out. So what we have here is a diagram of a cell. It has a number of pink chromosomes in its nucleus. Some section of the chromosomes contain genes. The entire cell of the chromosome is called genome. The chromosomes are made of the DNA, and DNA is made up of nucleotides. So what is the DNA damage? DNA damage is an alteration in a chemical structure of the DNA, such as a break in the strain of the DNA, a base missing from the backbone, or chemically changed base. Many things can cause DNA damage to occur. A lot of time, environmental factors lead to this damage. However, genetic disorder can also cause a DNA damage. Any kind of the DNA damage, if not reported properly, can lead to the serious health issues. So we know that the DNA damage can be caused by the environment or by a problem with our genetics. These factors can be simple, such as exposure to sunlight, or complex, such as a free radical generation that occur during the normal metabolic process. This picture shows different type of the factors that can cause a DNA damage, as well as different response that they induce. Cellular metabolism and a DNA replication error occur with our body and are what we would call intrinsic factor. UV light exposure, radiation exposure, and a chemical exposure are environmental factors that can cause DNA damage, and we would refer to them as extrinsic factors. When any of these things cause a DNA damage, they can lead to one cell cycle checkpoint activation, which would help you determine that cell has a damaged DNA and shouldn't reproduce. Second, transcriptional program activation. DNA repair, and if the DNA cannot be fixed, cellular apoptosis or cell death. So let's, like I mentioned, aerial DNA damage can lead to the disease if it is not fixed. Too much sunlight or UV exposure can cause skin cancer. Tobacco smoke or chemical exposure can cause a lung cancer. Free radical generation cellular metabolism can cause a mutation that leads to multiple types of the cancer. There are multiple kinds of the DNA damage, like any molecule. DNA can participate in a chemical reactions that change its structure. These changes are damaging because they are interfere with the DNA's normal function. Here are some examples of DNA damage. Both environmental and cellular factors can cause a double strain break, in which the breakbone of the both DNA strain and severed, resulting in a completely separate piece of the DNA. The next example, exposure to large amount of the energy, such as that from the UV light proceed by the sun, can cause two neighboring nucleotide bases to chemically attach to each other. Other chemicals within the cell can also attach themselves to individual nucleotides or actually connect the two strands of the DNA together. One set of these chemicals is reactive oxygen species, which have gained research attention due to their relationship to the both aging and cancer. Cells produce these molecules by byproducts when they make new cellular building block during stress response or due to the exposure to environmental sources like a UV light. One important DNA damage response is triggered by exposure to UV light. There are three categories of the solar UV radiation. Only UVA and UVB are able to penetrate Earth's atmosphere. Thus, these two types of the UV radiation are of the greatest concern to humans, especially as a continuing depletion of the ozone layer cause higher level of this radiation to reach the planet surface. UV radiation causes two types of the DNA lesion, and both of them lesion distort the DNA structure, introducing bends or kinds and therapy. Impeded transcription and replication. Relatively flexible areas of the DNA double helix are most susceptible to the damage. In fact, one hot spot of the UV induced damage is found within a commonly mutated oncogenes, which called the P53 gene. UV indeed DNA damage is repaired through the process known as a nucleotide excision repair. UV radiation induced DNA damage and its proper repair through nucleotide excision repair will be our main focus on this model, and the repair mechanism will be explained in the more detail in the following slides. So there is different repair methods for different DNA damage. Because there are many different types of the DNA damage, cells have multiple methods to repair all the damage. When a single nucleotide base is damaged, the cell uses a repair method called base excision repair. Because just the damaged base is removed and replaced. During the DNA replication, sometimes the wrong base is incorporated into the DNA strain. These type of the damage are repaired by the method called mismatch repair. Because there is a mismatch between the normal base pairing of the DNA molecule, the damage caused by the UV radiation, and carcinogenic like cigarette smoke is repaired by a method called nucleotide excision repair. DNA damage is inevitable, so DNA repair is necessary for the survival of the life on the Earth. Base excision repair fixes the damaged nucleotide bases. The nucleotide cytosine can easily degrade and lose a group of atoms called an amino group. This transforms the cytosine into a new base, uracil. Uracil cannot base pair with a guanine. This helps the cell identify the damaged cell. To repair damage, the uracil, the base is removed from the DNA backbone by the enzyme named glycosyl. Then the backbone is removed by other enzymes. The DNA polymorph enzyme fills the empty space with a new cytosine. Lastly, an enzyme named DNA ligase seals the DNA backbone and no evidence of the damage remains. Mismatch repair fixes the error left behind after DNA replication. The first step of this repair includes identifying the mismatch bases in a DNA strain, which is done by the enzyme mutease and mutual. Next, the enzyme mutual S determines which strain has the correct sequence. This is determined by the fact that the template strain of the DNA synthesis has methyl groups while the newly synthesized DNA strain doesn't have. The DNA backbone of the strain with a wrong sequence is cut into two places. A stretch of the DNA, including the incorrect base, is removed. Why there is such a large amount of the DNA removed? Methyl groups do not bind to every nucleotide base. Therefore, the DNA section between the closest methyl group and the faulty DNA base are removed. A DNA polymerase enzyme correctly fills the gap, and the DNA ligase enzyme seals the DNA backbone. UV radiation and other side and carcinogenics frequently cause two thiamine bases next to each other to bound together in a state of with the adenine bases on the opposite strain. So to repair this group of three enzymes working together, identifying the damaged bases, and precisely cut the DNA backbone on the either side of the damage, the three proteins are named UVRA, UVRB, and UVRC. The damaged fragment is removed. The layer of the single-strand DNA left behind is replaced by DNA polymerase. DNA ligase seals the DNA backbone, restoring the DNA to the original structure. So now we will form a short DNA strain. Our right arm will hold our nitrogenous base and our left arm will be the sugar base. Here is a view of our DNA strain from above. What would happen to our DNA strain after UV radiation? The UV radiation changed the chemical structure of the thiamine, causing them to bind to each other instead of adenine. The thiamine dimer introduced a kink in the DNA. How to fix that? Endonuclease remove the damaged DNA. But now there is a hole in our DNA strain. How is the fixed? What is needed? DNA polymers recruit new nucleotides to fill the gap. A ligase seal the new nucleotides seals the new nucleotides into the repaired DNA strain. And now we have a repaired DNA strain. But how do you mechanistically study DNA repair? How did these scientists figure out these details? One of the early scientific questions was, what are the necessary proteins to remove DNA damaged by the UV radiation? Research suggested there are particularly protein in a bacteria were necessary for the bacteria to repair UV damaged DNA, but no one had shown that no additional protein were necessary. Let's create an experiment performed by Dr. Aziz Sansar to see how the work is done. To perform an experiment, we need some DNA that has been damaged by UV radiation, and we need a purified protein that we suspect are involved in the repair process. The experiment will involve mixing the damaged DNA in the test tube with a different protein to determine which combination of the protein is able to remove the damaged region of the DNA. We need some way to determine if the damage has been removed. Likely DNA before and after the damage is removed have different properties, so which we can easily detect in the lab. So let's see one of the differences we can detect. So we can use the DNA gel electrophoresis to tell if the DNA damage has been removed. To do this, we put a drop of the DNA that has been combined with our purified proteins at one end of the slab of the gel. And then we apply an electrical current to the slab of the gel, which causes the DNA to move through the gel. The slab of the gel is like a very dense forest with so many trees and branches that a large animal, like an elephant, would have to move very slowly through this forest, but a small animal like a rabbit would easily find a path through. In this case, if a DNA is tightly wound up, like the damaged DNA is it moves through the gel quickly. If the DNA is stretched out, like what happened once the damage is removed, the DNA molecule move through the gel slowly. So using common DNA dyes, the DNA is in the gel can be seen as a bond with a naked eye. The damaged DNA will move faster, so when we look at the gel, the damaged DNA will be farther away from the starting point. This way we can tell if the damage is still in the DNA molecule or if it has been removed. There are two bounds of the DNA in most of the samples because there is a small amount of the DNA that is already relaxed. The intensity of the bond is directly proportional to the amount of the DNA present in each bond. In a small in sample 10, all of the DNA detected is relaxed, indicating that the protein UVRA, UVRB, and UVRC removed the cell, the damage relieving leaving only relaxed DNA. This proved that these three proteins are necessary and sufficient to remove UV damaged DNA. Now that we have learned about the DNA repair, let's learn how it's related to the cancer. We learned earlier that DNA damage, when unnoticed, can result in various disease states. Well, we also know that in many types of the cancer, DNA repair mechanisms are in fact damaged. This damage can be due to the genetic predisposition. So what does that mean? A genetic predisposition is a genetic characteristic that influences if a phenotype will occur under the influence of the pre-existing environmental conditions. Your genome is unique to you. Your genetic makeup in combination with your surrounding environment can influence if you are more or less likely to get disease such as cancer. Colon cancer, for example, is a disease in which genetic predisposition is observed. In 85% of the colorectal cancer cases, the initial mutation is in a gene called APC. The APC gene play a role in stopping tumor progression. So if you have a mutation in this gene, what do you think happens? The mutated gene is no longer able to effectively stop tumor growth. Therefore, cancerous tumor are able to grow unchecked. When the tamer forms, the cancer cell will then mutate the DNA repair mechanisms. So what happens now? What can doctors do to help individuals with a cancer? Conventional cancer treatments work by exposing cancer cells to toxin or radiation when they are most vulnerable to DNA damage. By purposefully eliciting DNA damage to these cells, the cancer treatment is promoting cell death. So let's look at an example. Here, the purple cell and a green cell represent cancer cell. A patient with colorectal cancer undergone conventional cancer treatment such as radiation therapy, and this results in the death of all of the purple cells. The green cells, however, persist. How can some cells don't die following a conventional cancer therapy? One possibility has to do with the DNA repair mechanisms. We just talked about it, how a mutation in the APC gene lead to cell turning of DNA repair mechanisms. Some repair mechanisms, however, are able to remain functional. This means that these cells have ways of repairing the damage that is caused by the cell cancer treatment. So they are able to survive and in time proliferate. So what do we do now? Researchers are now using the weakness to target this cancer cell or find a way to block the remaining mechanism in the cancer cell to stop or slow the growth. So let's go back to our patient. All the purple cells were effectively killed by the conventional treatment, but all the green cells remain. The green cells do not have mismatch repair or nucleotide excision repair. What repair mechanism is still functional? So what do we need to do? So thanks to modern research, we have a way to target this. So there are special way to target these cancer cells without harming your surroundings healthy cell or target therapy target. This means that the drug are not or less cytotoxic. By targeting these cells with these medical treatment, we are able to slow down or stop cancer cell from giving hopefully resulting in cancer remission. There are various treatments being developed to target the DNA repair mechanism with the hopes that they can be used in conjunction with the conventional cancer treatment. While some of these are in early study, others have already gone through some or all clinical trials. These are three drugs that have been FDA approved for a different type of the cancer. Lenioprasis is a type of the cancer therapy that target DNA repair mechanism in the cancer cell. This target PPAR, which is an enzyme involved in the DNA repair. The drug work by exploiting the DNA repair defect in the cancer cell. Lenioprasis has been FDA approved for use in some ovarian, breast cancer, and prostate cancer, but it is currently in clinical trials for the use in colorectal cancer. So if you go back to our patient that had undergone a radiation treatment, but had persistent cancer cell, the cell that maintain the base pair existing repair mechanism, we can now give them a medication that specifically targets their cancer cell with a functioning nucleotide excision repair mechanism. The idea is that the medication will lead to cell death and hopefully result in remission of the patient. This is the end of today's video. Thank you for attention and hope you will enjoy it.